Okay, this is the review for the lab final in Bio 9 for human physiology. It'll be 25 points, 25 multiple choice questions. We're going to do a quick review of the entire lab in the semester, or things that we feel important enough to be on the test. However, 10 to 15 points of this quiz will be on the last labs that we've done since the last quiz you've got, i.e. not cardiac, but urinalysis, kidney function, and respiratory. So, going back, let's start with homeostasis. Homeostasis is something we started right at the beginning. You're looking at variation around a set point, whether it be pulse rate by running in class, or glucose around the normal range, thyroid. Homeostasis generally will use a negative feedback loop. Negative feedback, the body senses a change, sends information to the integrating sensor, which causes an effect that negates that change. Some negative feedback systems would be body temperature, or if it's hormonal, it might be used insulin, the hormone insulin, to regulate sugar in the body. We're going to talk about acid, base, and pH. It's controlled through buffers, through respiration, or through the kidneys. Know what an acid is, having more hydrogen ions, or a base having more hydroxyl, and the pH is a measurement of that. Below 7 is acidic, above 7 is basic. Remember, the pH scale is a logarithmic scale, so every number change is actually a change of 10 times in the concentration. Below 7 acid, above 7 alkaline. pH scale 0 to 14. The buffer stabilizes the concentration of a solution. It can either donate or accept a hydrogen or a hydroxyl in order to turn a strong acid or a strong base into a neutral or a weak acid or weak base. The common one we see in the body is a carbonic acid bicarbonate system. You're going to have to know about diffusion, movement across a semi-permeable membrane, or a movement from a high concentration into a low concentration. Diffusion will move until the concentrations are equal. Rates depend on how much the difference is, the temperature, and how much surface area. Osmosis is diffusion across the semi-permeable membrane. Know about osmotic pressure. Know about the ionization. If you have a solute, the solid stuff, the solute versus the solvent, the liquid, that ionizes into two different particles, like sodium chloride ionizes into sodium and chloride, your osmotic pressure will double because you get the osmotic pressure of the sodium plus the osmotic pressure of the chloride. For osmosis, you've got a solute concentration difference on either side of the membrane, and the membrane must be permeable to the solution. Osmosis is the net movement of the water or the solute. Net movement of the water or the solution, not the solute solution. Know what a mole is. It's a gram atomic weight. Know how to calculate a molar or molal solution. Molarity, molar, is 1 gram atomic weight with enough fluid or solution to make 1 liter. Molality is 1 gram atomic weight plus 1 liter. That's what we normally use because it's more accurate. Osmolality, remember, it's your molality times your ionization. Tonicity is a relative term to another substance. meaning if it's more concentrated, it's hypertonic. If it's less concentrated, it's hypotonic. And if it's equal, it's isotonic. Remember, when you look at tonicity as in osmolality, you have to take in the ionization also. Here it's solutions of lower solute concentration are hypotonic, and water would be drawn in, but just like we did in the lab with the red blood cells in distilled water. The distilled water is hypotonic to what's inside. It moved into the cell, it lysed or burst the cell. And we did talk about red blood cells and do the red blood cell experiment in class. Osmolality, blood osmolality is in the hypothalamus, and it tells the kidney in the body whether you're thirsty and you need to take in fluid or you need to excrete fluid in there. Neurons. When you look at neurons, you're going to look at afferent and efferent. Afferent moving back toward the central nervous system, efferent moving away. Afferent would be a sensory neuron, such as touch, heat, light. Efferent would be generally your motor neurons. Okay, sensory receptors transduce energy or sensations into nerve impulses. And they go up into the central nervous system. You have mechanical, pain, chemo, and photoreceptors. Tonic and phasic, know the difference. Tonic is a receptor that does not stop firing as long as there's a stimulus. Phasic may adapt and stop firing, even though the stimulus might continue to be there. Phasic might be the clothes on your back or a smell that you get used to. Tonic might be pain, injury that you really don't want adapting. Law of specific nerve energies says that a receptor will only fire for its intended sensation. Hot, cold, light, pain, whatever. If you get that sensor to fire, it's still only going to fire for what it was intended. So if I stimulate a hot receptor enough to get it to fire by a pain, it's still going to fire as a hot. You're going to experience the hot, not necessarily the pain. Skeletal muscles have just an idea of the sliding filament theory and how muscles contract. We're not going to go into the anatomy of the muscle. You have flexors and extensors, agonists and antagonists. Biceps and triceps would be a classic example of that. Motor unit, know what a motor unit is and how it works. A motor unit is the neuron and however many muscle fibers it stimulates. A motor unit is an efferent, meaning it's coming away from the CNS. I have basic idea of how the sliding filament works. Calcium released from the sarcoplasmic reticulum after stimulation from the motor unit, causing the troponin and tropomyosin to release, causing the myosin heads to attach and getting a power stroke. That's very brief, but you have an idea how that works. The forming of cross bridges is after the troponin allows the tropomyosin to move. Calcium is released from the sarcoplasmic reticulum. Know the structure of a heart, the blood flow through the heart. know the circulation. Have an idea of what valves are where in the heart. Tricuspid in the right atrium, the right ventricle, bicuspid left atrium, left ventricle, pulmonary and aortic. Heart sounds. What a heart murmur is a defect in the valves, getting regurgitation. Know what the cardiac cycle is. Systole is a contraction, diastole is a relaxation. Okay, we talked about red blood cells. Know how to type. Know that an A cell has A antigen, B has B, AB has both, type O has neither. You're looking at a transfusion reaction. The person does have antibodies against the blood. You transfuse it, they're going to have a reaction. For instance, an A person has anti-B, a B has anti-A, O has anti-A and anti-B, AB has neither. Transfusion reaction, if you receive the wrong blood, the antibodies bind, they'll hemolyze the blood, can go into renal failure, they'll cause a lot of trouble. Compatible blood types, and O can only receive O blood, A can receive A or O, B can receive B or O, AB can receive any blood type. O is the universal donor, AB is the universal recipient. This is how the antigens work. Look at the abbreviation. RH is the antigen D. You're either RH positive or RH negative. We take that into account when we type. We try to give the same RH. In the mother may, if the mother is RH negative and the baby is RH positive, the mother may make antibodies to the RH on the baby's blood, and you get hemolytic disease of the newborn, or otherwise called erythroblastosis fetalis, where it's attacked across the placental membrane. Electrical activity of the heart, the sinoatrial node, down through the AV node, into the bundles of His, into the Purkinje system. So know about the contraction in general. Know how to look at an electrocardiogram, know what the waves are. P wave, what they stand for. Know what the waves stand for. Heart sounds we had earlier, lub, dub. Pulmonary function test. We did several. Know what the pulmonary function tests are. We did use the computers. We did a pulmonary function test. Lung volume and capacity. Tidal, expiratory reserve, inspiratory reserve, residual volume, vital capacity, total lung capacity, inspiratory capacity, functional residual. You know all these. Vital capacity is look at the relationships. Know what an FEV is. It's forced expiratory volume. And FEV 1, 2, and 3 is forced expiratory volume in 1 second, 2 seconds, and 3 seconds. It's used in diagnosing restrictive and obstructive lung disorders. Know what COPD is, asthma, and COPD, asthma, and emphysema. Kidneys, know what we did in the urinalysis, know what the tests are. Color would indicate know about the individual tests that we did and what they mean. You're looking at the kidney function, regulation of extracellular fluid. You've got the kidneys being controlled by the hypothalamus as to how much fluid is reabsorbed or excreted or urinated out. You have an idea of the nephron and how it works. The parts: glomerulus capsule, approximated convoluted tubule, loop of Henle, ascending and descending. The distal convoluted tubule, the collecting duct, and then into the bladder. We're not going to get into the anatomy of the glomerular capsule, know what a filtrate is, know how the kidneys can reabsorb water, and i.e. the countercurrent multiplier. Okay, know how the countercurrent multiplier works. What the ascending and descending loops of Henle do, and an idea of how ADH works, antidiuretic hormone. Aldosterone and renin-angiotensin system. Know what you do and did in your analysis. Know the color, how specific gravity, how concentrated. Go through the multi-sticks 10, the strips that we used. Know what each area looks for, i.e. you've got pH, albumin, blood, specific gravity, leukocyte, nitrate, total protein, and I think that's just about it. But go through that, know what each one of those things measures, and what you would see on that. And that is about it. That should cover most everything that you're looking for. Also on the kidneys, know what's excreted if it's high concentration. Know the lab on the kidney and the respiration. Know the details of the lab, exactly what we did during the lab. Thank you very much. That should take care of it.